My mission is to discover the language of nature, and I believe nature is intelligent.
There is a world under the earth, full of magic and mystery. It holds the consciousness of nature's connection to all living things.
You know, these mushrooms, they can heal you, they can feed you, they can kill you.
It's not like a vegetable, and it's not like an animal, but it's somewhere in between.
They support life, they convert life.
As you're walking, there's about 300 miles of fungi under every footstep that you take, and that's all over the world.
The bulk of the organism is growing underground, and it's composed of these long threads called a mycelium.
Almost everyone knows about the computer internet. The mycelium shares the same network design.
It's amazing what we don't know about mushrooms. They really are a frontier of knowledge.
What you just watched was actually a trailer for a film entitled Fantastic Fungi. I wanted you to see how big the topic of fungi is, and we will tackle only a minuscule amount of it today. We are zeroing in on medical mycology, specifically on superficial mycosis. I am Dr. Maria Caterina Latherin. At the end of the session, you, my dermatology residents, will be able to describe the clinical presentation of the 4 superficial mycosis, correlate the microscopic findings with the clinical presentation of the 4 superficial mycosis. You'll also be able to demonstrate proper specimen collection, processing, and reading of KOH wet mount. You'll be able to select the appropriate management for such infections, and you will be able to formulate measures for the prevention and control of superficial mycosis. What is mycology? By definition, it is a scientific study of fungi. It involves the study of its genetic and biochemical properties, its taxonomy, its use to humans, as well as its dangers. It comes from the Greek word myx, which literally translates to fungi. It has various beneficial as well as harmful activities, from decomposition, food and food production, and industrial production, to destruction of materials, including spoilage, allergies, and infections. Fungi are organisms with clearly defined cells that derives its nutritional requirements from complex organisms, specifically dead or decaying organic matter. As was mentioned in the clip, they are neither animal nor plant. There are 2 major groups of fungi, your yeast and your mold, and there are approximately 300 species that are pathogenic to man. They do have specific growth requirements, and these parameters are important if we are to try to grow them from collected specimens in order for us to be able to identify them. Yeast are unicellular, spherical to ellipsoid in shape, T to 15 microns. They multiply by budding and produce either blastospores or pseudohyphae. On culture, their colony are described as soft, opaque, approximately 1 to 3 millimeter in diameter. Molds, on the other hand, are multicellular filaments. On culture, they are described as having suede-like, velvety, fuzzy, or cotton-like surfaces of a variety of color and have tiny spores. The dimorphic fungi in its yeast form, which is the pathogenic form, are collected from tissues or exudates and need to be cultured in 37 degrees centigrade. The saprophytic or mold forms are found in nature and are cultured in 25 degrees centigrade. Molds can be characterized according to septations or color. The metaceous molds are pigmented or darkly colored. Fungal colonies are described according to texture, topography, surface and reverse colony, colors, and according to production of diffusible pigment from colony into the growth medium. Here's the taxonomy of the kingdom fungi. So for superficial mycosis, they are generally considered only as cosmetic, as they involve only the skin or the hair without invasion of living tissue. Therefore, there's no inflammatory response from the host. We will go through the 4 important superficial mycosis. First is herpetirizis versicolor, which is a chronic mild infection of the stratum corneum. Your causative agent is the Malassezia species. Depending on the skin color of the affected individual, it may present as white, pink, fawn, or brown. They are well demarcated, often coalescing patches covered with thin furfuraceous scales. Most commonly affected sites are your chest, upper back, arms, and abdomen. When you shine it with wood's light, it would fluoresce pale green. The Malassezia furfur is a lipophilic yeast that is dimorphic and is actually part of the normal flora of human skin. It may, however, also cause Pityrosporum folliculitis and is associated with seborrheic dermatitis. Other factors that are implicated in the growth of Malassezia furfur are heat, humidity, oily skin, hyperhidrosis, heredity, and immunodeficiency states. Your specimen should be skin scrapings collected either by scalpel blade, glass slide, or tape stripping. This should be processed with 10% KOH, glycerol, and Parker ink for better visualization. Under the microscope, you would see thick-walled cells that are oval or round and produce buds. They also have pseudohyphae and spores, producing what they call spaghetti or ziti and meatballs, with the pseudohyphae as your spaghetti or ziti and the spores as your meatballs. You treat this either topically or orally, depending on the amount of surface area involved. The second superficial mycosis is your tinea nigra. Again, they are superficial and asymptomatic since it affects only the stratum corneum. It is caused by your Hortea werneckii, and the lesion in the skin would be dark, brown to black, discoloration with little or no scaling. It is usually a single lesion and more commonly affects the palms and the soles. Hortea werneckii is dimorphic, able to tolerate high salinity, and contains melanin producing its brown color. Under the microscope, you'd see olive brown, multiple branched septate hyphae, and elongated budding yeast cells with melanized cell walls. The terminal portion is usually hyaline. In nature, they are found in soil, wood, vegetation, paint, and even shower curtains. Treatment consists of keratolytic solutions or azoles. The third superficial mycosis is pedra or white pedra, derived from the Spanish word for stone. It is a superficial infection of the hair strand. So you'd expect to see this on the scalp, eyebrows, beard, axilla, and groin. It is transmitted by direct contact, and you'd see small soft nodules which are adherent to the affected terminal hair. As the name implies, it is white or cream-colored. Since the hair shaft is weakened by this infection, you'd expect to see cut-off hair strands. White pedra is caused by your Trichosporon bagelii, which produces both arthroconidia and blastoconidia. The colonies would appear as white or yellowish to deep cream-colored, smooth, wrinkled, velvety, and dull in appearance with a mycelial fringe. We treat this by shaving all of the infected hair. We may try cotrimazole cream if there is minimal involvement. In the book, there is mention of amphotericin B lotion, which is unfortunately not available in the Philippines. The last superficial mycosis is your black pedra. It again affects the hair, and you'd see 1 to 2 millimeter firm, hard, adherent nodules along the length of the hair shaft all the way down to the scalp. In some cultures, they consider this as a mark of beauty and prestige. The nodules are composed of ascostromata, or fruiting bodies of the fungus, which contains ascus and ascospores. Black pedra is caused by Pedrea hortei, which have darkly pigmented branching hyphal strands with irregularly shaped swollen cells. They have ascostromata, asci, and ascospores. On culture, the colonies are slow-growing, small folded with velvety surface, and are dark brown to black in color. They may produce a reddish-brown diffusible pigment, and the reverse is usually black. Treatment usually involves cutting of the affected hair, use of keratolytics, and terbinafine may actually work. We now have a video on skin scraping and KOH wet mount.
This is a video on how to do a KOH scrape and prep. So you're going to use 2 glass slides for this. The first glass slide is going to be placed firmly below the lesion that you want to scrape. The second glass slide you're going to hold between your thumb and your index finger, and you're going to hold it perpendicular against the skin and scrape firmly, but not too firm as to draw blood, very quickly, like this. And now you see the scale is on the slide. Now that you have the scale on the slide, you can use the other slide to organize it so that it will all fit underneath one cover slip. Like this. Next thing we're going to do is take the KOH. You can use either 1 or 2 drops depending on how much scale you have. Next thing we're going to do is place the cover slip on over the KOH. So you're going to do this at an angle to prevent bubbles. So we're at an angle, and then we slowly lower the cover slip down, like so, to prevent bubbles. This is what it looks like once you have the cover slip on. Some people like to use a flame to help destroy some of the scale. You'll pass the flame underneath just a couple of times to heat it up without boiling it. Like that. And if there's any char underneath the glass slide, you can use some alcohol to clean it off to make sure you get good visualization with the microscope. If there's any char from heating up the bottom of the slide, you can simply use an alcohol pad to rub the bottom of the slide and remove the char to make sure that you have good visualization with the microscope. Okay. Of course, you should be wearing gloves for the whole procedure. The next step is to take the slide and put it on the microscope. Then we're going to center the area with the cover slip underneath the lowest power. So you need to make sure that you're on the lowest power. Next step, turn on the microscope and position it so that the area that you think has the most concentrated amount of scale is underneath the light. Now we found the scale on low power, so we're going to move to medium power. And there's right in the middle is the area that we're looking for. Now we're going to move to high power. And as you can see, we need to do a little bit of focusing. So there, you can see all the little lines. Those are the hyphae that we're looking for.
Go back up out of focus.
So now we're out of focus. We're going back down towards focus.
The first thing that comes into focus are the hyphae. Go down lower into focus. And you're going to see the outlines of the keratinocytes next. And now
as we go back up, there's the hyphae again.
Back up more, and back down. So you can see that the hyphae come in and out of focus. Fantastic. So this is a positive KOH.
In summary, we have your 4 superficial mycosis, namely pityriasis versicolor, white pedra, tinea nigra, and black pedra, which affect only the stratum corneum, are non-inflammatory and therefore asymptomatic, and basically can be treated by shaving or cutting the hair and by the use of your topical azoles and keratolytics. Thank you, and I hope you learned something new today.